The shroud has some very interesting characteristics, and these are not only interesting, but they're scientific characteristics.
For over 600 years, no one has been able to resolve the debate over the Shroud of Turin. That is why I am not interested in who is right or wrong, but rather in what the cloth itself is telling us. Scientists once believed they had solved the problem. Radiocarbon dating, 14th century, medieval forgery, case closed. So when a research team compiled all the DNA data, pollen, blood analysis, and fabric spectroscopy and fed it into Grok AI, they expected the machine to confirm what the textbooks had always taught. It refused. The answer it returned was so far beyond all predictions that the room fell silent. Because if the AI is right, this cloth has been to places history claims it never reached, and the blood on it belongs to a man who died in a way that modern forensics can read in precise detail, in a language that no brush and no artist could ever create. So what did it see? Stay with me, because once you realize this, you cannot unsee it. Along the banks of the Po River in northern Italy, inside the Cathedral of St. John the Baptist in the city of Turin, there is a specially designed silver box. Inside that box is a rectangular linen cloth woven in a 3-1 herringbone twill, measuring 4.37 meters long, 1.11 meters wide, and a mere 0.34 millimeters thick. The kind of artifact a tour group might glance at for 5 minutes before walking away. But the moment you step closer, that sense of normalcy vanishes, because on the surface of the cloth, under normal light, is the faint brown image of a man, both front and back, full body. The man is bearded. On his head are marks consistent with something like a crown of thorns. On his wrists and feet are wounds. On his back is a series of lash marks consistent with a brutal scourging. On the side of his chest is a massive wound. And here is the part that makes you stop. No paint, no pigment, no dye. The image was not painted onto the fabric. It appears to be embedded directly into the outermost fiber layer of the surface, at a depth so incredibly thin that even the most powerful microscopes struggle to explain how it was formed. This is no ordinary painting, and for over 600 years, no one has been able to explain what it is. The first time the cloth clearly appeared in history was around 1353 in France, in the hands of a knight named Geoffrey de Charny. From there, it moved to Liry, then Chambéry, and then to Turin in 1578, where it remains today. Millions of people have visited it during its rare public exhibitions. The Catholic Church has never officially declared the shroud to be authentic. A cardinal in the 1980s described it as a historical icon rather than a verified relic, but the Church has never rejected it, either. The reason is simply that science cannot debunk it either. No other relic in history has ever been subjected to so much scientific testing. And every debate, every experiment, every peer-reviewed paper all revolves around the exact same question. How old is this cloth, and how was the image on it formed? For a very long time, textbooks offered a stable and comfortable answer. In 1988, samples were cut from the shroud and sent to 3 independent laboratories, Arizona, Oxford, and Zurich. Each lab used accelerator mass spectrometry to measure the carbon isotope ratio in the fibers. The results from all 3 labs pointed to the exact same time frame, between 1260 and 1390 AD, with 95% confidence. Medieval, not ancient. The announcement was widely reported around the world. Many concluded the debate was over. The shroud is a fake, case closed, time to move on, and the C14 result did not stand alone, it had allies. Almost 600 years earlier, around 1389, Bishop Pierre d'Arcis in Troyes, France, sent a letter to Pope Clement VII, claiming that an artist had confessed to painting the image on the cloth himself. A direct confession from the forger himself, hard to ignore. Then came Walter Macron, a renowned American microscopist, who examined fiber samples from the shroud under a polarizing microscope in 1979. Macron claimed he found billions of submicron pigment particles, red ochre and vermilion, in a collagen tempera binder. That is the exact popular paint formula of the 14th century. According to Macron, the cloth was simply a masterful painting, created by a medieval artist right before its first recorded historical appearance in 1356. On paper, this theory held up. 3 pillars pointed to the same answer. The C14 result said 14th century. The bishop's letter said someone confessed. Macron's analysis said there was paint pigment. 3 pieces of evidence from 3 independent sources, centuries apart, all agreeing with each other. Put enough evidence on the table, give it enough time, and even a 600-year-old starts looking solvable. It was a fair argument, and it deserved respect. The trouble only started when another group of scientists stopped debating the theory and decided to actually test each pillar one by one. The first pillar collapsed sooner than you might think. In 1898, an amateur Italian photographer named Secondo Pia was granted the privilege of photographing the shroud during a public exhibition. Photography back then meant heavy equipment, large glass plates, and manually developing chemicals in a darkroom. Pia set up his camera in the cathedral, exposed 2 glass plates, and brought them back to his darkroom. In the dim red light, he lowered the glass plate into the chemical bath and waited. As the image slowly emerged, his hands began to shake. Normally, a photographic negative reverses tones and looks distorted. But what Pia saw was the exact opposite. It was more real than the original. A face appeared with stark clarity, three-dimensional, deeply detailed, like a direct photograph of a real face, rather than a negative of a painting. Eyes closed, a scar on the nose, a beard, the expression of someone who had endured something colossal yet remained at peace. Pia almost dropped the glass plate. This meant someone had created a photographic negative image on cloth at a time when photography did not even exist. No one could explain how. From then on, for over a century, the cloth was tested using X-rays, ultraviolet, infrared, laser scanning, and spectroscopy, each test revealing something new, each answer spawning a new question. Then in 1978, the STIRP project brought over 30 scientists into Turin with 80 crates of equipment, examining the cloth continuously for 120 hours without a break. The results, published in 20 peer-reviewed papers, directly shattered Macron's pillar. Chemists Heller and Adler confirmed the red stains contained hemoglobin, serum albumin, and bilirubin, real blood, not paint. They discovered that the fabric fibers in the image area had been chemically altered only on the outermost 200 nanometers of the listen fibers, causing the brown discoloration visible to the naked eye. No pigment, no binder, no brushstroke directionality. The image penetrates only 0.2 microns, 350 times thinner than a human hair. No known painting technique could create this. Then in 1976, two U.S. Air Force physicists fed photographs of the shroud into a VP8 image analyzer, a device that converts brightness into vertical relief. Normal photographs and paintings fed into it yielded distorted results. But the image on the shroud produced an anatomically precise, three-dimensional relief. The image had encoded 3D spatial information before computers even existed. STIRP's final report in 1981 stated a sentence that remains unrefuted to this day. The image on the shroud is that of a real human form of a scourged and crucified man, not the product of an artist. But no combination of physical, chemical, biological, or medical elements can adequately explain the mechanism of image formation. The scientific community did not accept the 1988 C14 results as the final verdict because the sampling methodology revealed severe flaws. The biggest issue lay in the location where the sample was cut. The 1988 sample was taken from an edge corner of the cloth, precisely the area that had been handled the most over centuries of public displays, restorations, and inspections. Subsequent chemical analysis discovered that this corner region had been repaired at some point. New threads were woven into the old fabric, creating a hybrids zone containing both medieval material and potentially ancient material. Hold on tightly to this detail because it dictates everything. Radiocarbon dating measures the ratio of carbon isotopes in a sample. If the sample is a mixture of ancient original fabric and medieval repair material, the result will reflect that mixture, appearing younger than the original cloth. Some researchers estimated that even a small percentage of new material woven into the old threads could skew the apparent age by centuries. The 1532 Chambéry fire, when molten silver dripped onto the folded cloth, could also have altered the carbon content within the fibers. Then in 2019, historian Tristan Casabianca obtained the original raw data from the laboratories through a legal request sent to the British Museum, Oxford, and Arizona. Upon reanalysis, he discovered significant statistical heterogeneity among the labs. The ages of the subsamples varied monotonically according to their spatial location, suggesting there was an unremoved contaminant distributed unevenly. In 2020, physicist Paolo Di Lazzaro and his colleagues published their own statistical analysis, concluding that the 1988 results failed to meet current precision requirements. At the 2025 International Conference on the Shroud of Turin, Casabianca presented further new documentation revealing questionable methodological adjustments during the dating process. But the debate is still far from settled. A 2026 study in the Nature journal NPJ Heritage Science analyzed fabric pieces from the 1988 sample and concluded they were consistent with the main body of the shroud. The 1988 results are therefore viewed by a significant portion of the scientific community as inconclusive rather than definitive. Not because the labs made mistakes, but because the sample they tested may not have been representative of the original cloth. The three pillars, Macron, the bishop's letter, the C14 dating, now all three were shaking. When radiocarbon dating was no longer deemed reliable, a research team at the University of Padua in Italy decided to ask an entirely different question. In 2015, they launched one of the most ambitious scientific investigations ever conducted on the shroud. Their goal was not to prove or disprove authenticity. Their goal was to use DNA to trace the cloth's journey, to figure out where it had been, who had touched it, and what the fabric had silently collected over centuries of movement and contact. They utilized specialized, ultra-clean equipment to collect microparticles from the surface and from between the threads of the cloth. The process was incredibly meticulous because contamination is the greatest enemy of ancient DNA analysis. Any modern particle, a flake of skin from a contemporary researcher, could ruin the ancient signal. The team worked under strict control protocols, using vacuum systems with ultrafine filtrators, and analyzed particles that had also been collected during previous restorations. They then applied next-generation sequencing, NGS technology, capable of simultaneously reading millions of tiny genetic fragments and reconstructing the patterns they represent. Human mitochondrial DNA, plant DNA, animal DNA, all were extracted from various regions of the cloth. Then they compared this entire dataset with global genetic databases representing populations from all over the world. And here is the part that changes everything. The researchers compiled all the DNA results from the University of Padua along with the pollen data from Max Frey in 1973 and Avenome Danon in 1997, combined it with fabric analysis data and blood profiles, and fed it all into Grok AI's analytical system. The AI was not told that this was the Shroud of Turin. It was not told that the cloth was connected to religion, to the crucifixion, or to any desired conclusion that anyone might hold. Its sole task was this: compare these biological traces with genetic databases, geographic distributions, and historical timelines, and then state which picture fit best. It only knew haplogroups, plant species, chemical markers, and millions of comparative data points. Humans entering this problem brought along their beliefs. They expected a forgery, or they expected authenticity, and they tended to see what they expected. The AI only knew patterns, frequencies, and distributions. Then the report came out, and a picture emerged that no one on either side was prepared to accept. If the shroud had been produced by a forger in France or northern Italy around 1350, the genetic traces on the cloth would be dominated by Western European DNA. France, Italy, maybe a little broader European variants from pilgrims who visited it during the 200 years it spent in Europe before the DNA study was conducted. And indeed, European DNA is present on the cloth. That was expected. But European DNA is just a small piece of the puzzle. When the AI finished processing all the data from the 2015 Barkecha study, the picture that emerged was vaster than anyone predicted. Researchers identified genetic markers linked to Middle Eastern populations, specifically markers matching the Druze community, a population that has lived in the mountainous regions of Israel, Jordan, Lebanon, and Syria for centuries with very little outside interbreeding. The relative isolation of this community makes their genetic profile so stable and distinct that geneticists consider the presence of the Druze marker to be a strong pointer to the Levant, exactly the region where Jerusalem is located. The cloth bears a marker pointing straight to where the crucifixion story began, but it does not stop there. Genetic traces linked to North Africa and East Africa are also present, markers associated with Egyptian and Ethiopian populations, regions with significant early Christian communities. Then came markers from South Asia, markers common in the Indian subcontinent, and most shockingly, traces related to East Asian populations, including markers linked to Chinese genetic lineages. Pause here for a moment. A cloth that researchers initially assumed might be a European medieval forgery instead carries DNA from China, India, East Africa, the Levant, and Europe. The question that any honest scientist must ask when faced with this result is simple. A forger working in France around 1350 would have exposure to Western European DNA. Perhaps limited exposure to Mediterranean peoples via trade routes. But genetic material from India and China on a cloth produced in 14th century France is not something normal medieval European life leaves behind. These are not regions that a French artisan would casually encounter. These are regions connected by the Silk Road, the ancient trade network linking Europe with the Middle East, Central Asia, South Asia, and China throughout the first and second millennia. The DNA pattern on the shroud matches the geographic footprint of the Silk Road far more closely than the footprint of medieval France. And when the AI overlaid the DNA map onto the hypothesized historical journey, Jerusalem to Edessa, Edessa to Constantinople, Constantinople to France, France to Turin, the genetic traces did not lie scattered randomly. They were layered, one century stacked upon another, in exactly the sequence an object would collect if it had truly traveled through the most internationally connected cities of the pre-modern world. Then Grok AI moved on to the blood, and this is where everything completely changed. For years, some researchers argued that the red stains on the shroud could be paint, ochre, vermilion, or medieval red pigments. But when chemists Heller and Adler examined it using independent analytical methods, they confirmed the presence of hemoglobin, serum albumin, and bilirubin, not paint, real human blood. type AB blood, relatively rare, found in only about 3% of the global population, but significantly more common in Middle Eastern populations. Hold tightly onto that detail. But the chemical composition of the blood is the part that truly gives everyone pause. Inside the blood, researchers found unusually high concentrations of creatinine and ferritin nanoparticles. These are not substances associated with a normal death. They appear in high concentrations in the blood of someone subjected to severe physical trauma, torn muscles, massive blood loss, profound dehydration, the systemic collapse of the entire body. The medical term for this pattern is rhabdomyolysis, a condition that occurs when muscle tissue is so severely damaged that it releases its breakdown products directly into the bloodstream. The blood carries clear characteristics of post-mortem blood. Potassium had migrated out of the red blood cells into the plasma, creating a hyperkalemic state that only occurs in highly acidic blood after death. The serum and red blood cells had separated, a forensic phenomenon that only happens post-mortem, never in fresh wounds. There were traces of pulmonary fluid, consistent with pulmonary edema due to respiratory failure and extreme trauma. Wound analysis identified evidence consistent with over 100 distinct injuries, a pattern researchers compared to Roman scourging. Then comes the detail that most people overlook, but which dictates everything. The blood flow streams on the cloth run in multiple different directions. The body was moved several times after death, exactly matching the description of a body being taken down from a cross and laid into a tomb. And beneath the bloodstains, the yellow discoloration forming the body image does not exist. The blood got onto the cloth first, and the image formed later, without damaging the bloodstains. When the complete blood profile was fed into Grok AI alongside the DNA and pollen data, the system synthesized a chilling conclusion. If this was a forgery, its creator would have needed detailed forensic knowledge of post-mortem blood degradation, serum separation, muscle trauma biochemistry, and the markers of rhabdomyolysis. That knowledge did not exist until the 19th century. An artist could paint the shape of a wound. An artist could smear red paint onto cloth to mimic blood. But no artist could use paint to recreate the specific chemical profile of blood produced inside a body undergoing catastrophic trauma. Chemical signatures identifiable only by microscopes and analytical equipment that did not exist in the 14th century. The blood tells one story, but the cloth itself tells another. Up to this point, everything has revolved around what lies on the cloth: DNA, blood, pollen. But the greatest mystery is not what is stuck to the surface. The greatest mystery is the image itself. And this is the part where everything starts to fall apart. Remember, the image on the shroud was not painted. There is no pigment. There is no binder. There is no brushstroke directionality. The brown discoloration visible to the naked eye rests solely on the outermost 2-3 fibrils of each thread, penetrating exactly 0.2 microns, 350 times thinner than a human hair. The colorant is conjugated carbonyls, the result of the accelerated aging and oxidation of the cellulose structure right at the linen fiber's surface. Not any substance added from the outside. The fabric itself has been altered, as if something touched its outermost layer and changed its chemical structure without penetrating any deeper. The image is a photographic negative, created before photography existed. It encoded three-dimensional information before computers existed. And to this day, no one in the world has been able to replicate it. When the AI was asked to compare all known painting techniques from ancient to modern times, tempera, fresco, encaustic, printing, thermal transfer, airbrush, digital, not a single technique could simultaneously produce all of these characteristics. Not a single one. Then came the modern experiments. In 2011, physicist Paolo Di Lazzaro and his research team at ENIA, the Italian National Agency for New Technologies, Energy and Sustainable Economic Development, used an excimer laser emitting in the vacuum ultraviolet range to irradiate raw linen. They achieved a superficial discoloration similar to the image on the shroud, so similar that it was difficult to distinguish even under a microscope. But there was a problem. They could only produce it over an area of a few square centimeters. In 2018, the European Synchrotron Radiation Facility published a study confirming that, theoretically, the image could be replicated by an ultra-intense, ultra-short flash of ultraviolet radiation like a laser pulse. But the energy required to produce that effect uniformly across the entire body image, both front and back, on a 4.3-meter-long cloth, would far exceed the capabilities of any medieval artisan. And here is the part that makes it all so hard to swallow. It also far exceeds the capabilities of modern technology. Not just by a little, by a complete magnitude. When the AI synthesized all the physical and chemical data of the image, it delivered a conclusion that no one wanted to hear. The image was not painted, not printed, and not burned onto the cloth. It looks most like the result of some form of radiation, yet it does not match any known natural radiation source. Some researchers have proposed that the energy required to create the image might represent something that physics cannot yet fully explain. They do not say this out of religious intent. They say it as scientists. And that statement still stands in scientific publications unchallenged because no one has offered a better explanation. If no one knows how the image was formed, perhaps the answer lies where no one thought to look right under the surface of the fabric, in pollen grains that have lain dormant for thousands of years. In 1973, Swiss criminologist Max Frey was permitted to take dust samples from the surface of the shroud. Within those samples, he identified 58 distinct plant species through pollen. Some came from European flora, consistent with the cloth's history since the 14th century. But the majority came from plants native to the Middle East, Turkey, and the Anatolian region. Among them, 22 species are found only in the regions of Constantinople and Edessa, and 7 species only grow in Israel. In 1997, Israeli botanist Avinoam Danon confirmed and expanded upon these results, identifying 28 pollen species and plant images, many of which only grow around Jerusalem and bloom from March to April. One species attracted particular attention, Gundelia tourniforte, a desert shrub with long, rigid thorns strong enough to be woven into a circular shape. Its pollen is present in massive quantities on the cloth, heavily concentrated particularly around the head and shoulder regions of the human figure. Researchers familiar with ancient Roman practices note that the thorns of this plant are physically perfectly capable of being fashioned into a crown of thorns like the one described in the Gospel accounts of the crucifixion. Its pollen blooms in early spring, around the time of Passover. Another species, Zygophyllum dumosum, which only grows in a strictly limited geographical area. The Judean Desert and the Sinai Peninsula is yet another botanical pointer aiming straight at the same region. In 2015, Cagliari and Cannavaro added to this with microparticle analysis, confirming not just pollen but linen fibers, cotton fibers, mites, fungal spores, minerals, and particles associated with bodily fluids. And here is the crucial argument you need to hold on to tightly. Pollen cannot be applied with a brush. It cannot be mixed into paint. It is only deposited naturally when a plant blooms nearby, or when a person or object that has come into contact with the flower touches the surface. A forger working in 14th century France would have no way to access pollen from plants that only grow in the Judean Desert between Jerusalem and Jericho. The pollen on the cloth in France points specifically to plants that bloom in Jerusalem in the spring. This is not a detail a forgery can explain, but only physical presence in that region can. When the AI overlaid the pollen map onto the DNA map, 2 completely independent datasets pointed to the exact same geographical journey. The DNA says the cloth traveled from the Near East through Anatolia to Europe. The pollen says the exact same thing. In the city of Oviedo in northern Spain, over 1,000 kilometers from Turin, inside the Cathedral of San Salvador, there is another cloth. It is much smaller, only about 84 by 53 centimeters, a linen face cloth bearing no visible image, just massive bloodstains. It is called the Sudarium of Oviedo. And according to tradition, this is the cloth that was placed over the face of the same man whose full body was wrapped in the Shroud of Turin. The history of this cloth can be traced back to the 7th century, 700 years earlier than the first recorded appearance of the Shroud of Turin in France. 2 cloths, 2 countries, 2 completely separate histories of custody. And when the AI placed their data side by side, the results changed everything. The Sudarium bears the exact same blood type, the rare type found in only 3% of the global population. The probability of 2 random objects sharing a rare blood type is already low. But that is not the scary part. The scary part is the geometry. Independent forensic studies superimposed the bloodstains on the Sudarium over the bloodstains on the facial region of the Shroud of Turin, and they matched perfectly. Not a rough match. A geometric match, the same locations, the same distribution patterns, the same flow directions. 2 cloths in 2 different countries, with completely distinct custodial histories across centuries, bear biological traces that match to a degree that modern forensics can confirm. Now think about this very carefully. If the Shroud of Turin was a medieval forgery, you would already need a forger possessing knowledge far ahead of his time. But if the Sudarium is also a forgery, you would need 2 distinct forgers in 2 different countries, hundreds of years apart, who somehow coordinated the bloodstains so precisely that modern forensics, using equipment neither forger could even imagine, confirmed they came from the same face. Or there is a much simpler explanation. Both cloths once touched the exact same man. When the AI compiled the data from both cloths, blood type, bloodstain geometry, chemical profiles, it categorized them as having a high probability of originating from the exact same biological source. 1 cloth you can doubt. 2 cloths telling the same story, that is a pattern. And this is when the AI did what no human expert wanted to do, it put everything on the same scale. If the 1988 C14 results were right, and the cloth truly originated in the 14th century, then the forger must have simultaneously accomplished all of the following. Used real post-mortem blood with precise forensic characteristics, potassium having migrated from the red blood cells, serum having separated, creatinine and ferritin at the high concentrations typical of catastrophic trauma, before forensics even existed. Created over 100 wounds in a pattern precisely matching Roman scourging, before Roman archaeology existed. Created a negative image on cloth before photography was invented. Encoded three-dimensional information into the image before computers were invented. Confined the image strictly to 0.2 microns of the fiber surface by oxidizing the cellulose without leaving behind any pigment whatsoever. A technique no one knows how to replicate even today. Placed pollen from Jerusalem and Anatolia onto the cloth in France. Coordinated bloodstains with another cloth sitting in Spain so accurately that modern forensics confirm they match. And did all of these things at the same time on the very same cloth, without leaving a single flaw that over a century of modern science could detect. The AI evaluated the probability of a 14th century individual simultaneously possessing all of the above knowledge and technical capability as infinitesimally low. But here is the paradox, and it is a genuine paradox, not a rhetorical trick. Because if the cloth is not a forgery, if it truly wrapped a corpse 2,000 years ago, then the mechanism of the image formation still remains outside all known scientific explanations. Vacuum ultraviolet radiation is the closest candidate, but no natural radiation source produces enough energy to imprint the image across the entire cloth without burning it. Both conclusions lead to the impossible, a forgery impossible, authentic impossible. And that is exactly what sets the Shroud of Turin apart from every other mystery in history. It is the only question where both answers are impossible. None of these findings prove that the shroud is what believers claim it is. They were not designed to do that. Science does not prove miracles. But these findings, taken together, have prevented science from debunking the shroud in the way many hoped it would when the laboratory doors first opened. The AI did not solve the mystery. It did not pick a side. It did something much worse. It made the mystery clearer, sharper, and harder to ignore than ever before. DNA tells the story of a cloth that traveled through the most interconnected cities of the ancient world. Pollen points straight back to Jerusalem in the spring. The blood bears the chemical profile of a violent death that modern forensics can read line by line. The image exists at 0.2 microns on the fiber surface through a mechanism no one can replicate. And another cloth, in another country with a separate history, is telling the exact same story. The cloth remains what it has always been, an object posing a question that no one has been able to fully answer. The Catholic Church has never officially declared it to be real. The Church has never declared it to be fake, either. Its last physical exhibition was in 2015. In 2025, there was only a digital exhibition, and the DNA evidence extracted from its threads in 2015 did not close the question. It deepened it, stretching it back across 2,000 years of human contact, human history, and human longing. And the researchers who ran that data are still thinking about what they found. So I want to hear from you. After everything we have just gone through, what do you think this is? A forgery by an artistic genius ahead of his time who possessed the knowledge of 6 centuries into the future? A true relic with a formation mechanism that science has yet to grasp? Or something else entirely for which we do not yet have a name? Let me know in the comments section. I read them, and the most compelling theories often come from you guys. And if you believe that history is still hiding much more than it tells, hit subscribe and ring the bell so you can be the first to know when the next discovery refuses to stay buried.